Okay, now we're going to talk about physical methods of microbial control. First, we're going to start with the temperature, all right? And temperature-mediated methods can be broadly divided into two subcategories. Look, you can have moist heat, okay, and you can have dry heat. So let's talk about each individual group separately, and then we're gonna add some more considerations. So in the moist heat group, what you have is autoclaving. So this is the most effective method of microbial control known to us so far. It is sterilizing. Does that make sense? So it's high pressure steam, pressurized steam. And this is what I want to focus your attention on. I'm going to focus mostly on, what level of control can we achieve? Can we sterilize, or it's only disinfecting? Okay, I promise I will tell the important stuff. Am I clear? Autoclaving. Then we have boiling. So boiling is not sterilizing; it is disinfecting method. Right. So it does not destroy bacterial endospores, the most resistant structures, right? And then we have pasteurization. It's also considered to be disinfecting. It just got its own name, okay? So it removes most zoonotic microorganisms that cause diarrheal diseases such as E. coli, Campylobacter jejuni, various species of mycobacteria, Coxiella burnetii, all kinds of pathogens that can be found in the products like milk, poultry, eggs, wine, all that kind of stuff. Does that make sense? Are we clear here? Okay. And what about dry heat? For dry heat, we have incineration, obviously sterilizing method. Incineration is a glorified word for burning. And then we have desiccation, also known as dry oven methods. So in the table that you have in the notes, it says that it's sterilizing method. It's not reliably sterilizing. Am I clear? It's not reliably sterilizing. So we're going to put disinfecting. Okay. Now, so far we good? Now what about, so these methods, so let's compare moist heat and dry heat. So I'm going to use our newly learned parameter, decimal reduction time, okay? So d-value. So d-value for moist heat versus d-value for the dry heat. So listen, at the same temperature, for the same microorganism, d-value for the moist heat will always be lower, significantly lower, okay? So moist heat is more effective. Does it make sense? moist heat is more effective, therefore D value for moist heat is lower, kills it faster. Couple of heat-specific terms that I want you to know. So, thermal death time and thermal death point. Okay, I'm not going to write the definitions, I'm going to say them. There are definitions in the notes. Listen, what's thermal death time? Time that is required to destroy all signs of microbial life at the given temperature. Does that make sense? So if we say that for Staph aureus thermal death time at 121 degrees Celsius is 20 minutes, means that for Staph aureus, for that particular microorganism, it takes 20 minutes to kill everything at 121. Thermal death point is the temperature which is required to kill all signs of that particular microorganism in 10 minutes. Okay? So thermal death point is the temperature that is required to kill all cells of that particular microorganism in 10 minutes. So obviously, let's say we compare endospores versus bacterial cells. For endospores, thermal death point will be much higher because you will need, I don't know, like 300 maybe degrees Celsius with a steam to kill all endospores in 10 minutes. Am I clear?
Does it have to be 10 minutes or is that just...
For definition of thermal death point, yes, it is like 10 minutes. So let's say we compare Bacillus subtilis with E. coli, and I tell you that for Bacillus subtilis, thermal death point is 150. For E. coli, it's 100. So you think, okay, in order to kill all of it in 10 minutes, for Bacillus, I need much higher temperature, which means Bacillus is much more resistant. Does that make, again, it's just another way to compare the resistance of different microorganisms, okay? Now, sort of cold methods. Again, you see, it's really hard to try and do the proper, like, absolutely strict classification. So we're trying to get the best of it. So, they are bacteriostatic. Kind of obvious, refrigeration and freezing. Does that make sense? Both of them are bacteriostatic or virostatic or fungistatic. Now, typical question, does freezing kill microorganisms? Yes, it does, but we can't rely on repeated freeze-thaw cycles to obliterate all microbes. Does that make sense? So this is why we consider cold methods of microbial control static rather than cytal. Are we clear so far? Good, awesome. I'm trying to think what I forgot. I probably forgot something. I can't think of what I forgot here. A little, well, I kind of put it under cold methods too. It's called Glyphilization. It's freeze drying. Glyphilization is actually used to preserve microorganisms. Those cultures that you worked with today, the ones in the tubes, they technically can be lyophilized, so bacterial cells can be freeze-dried and saved for the posterity. Okay, we good so far? Make sense? Okay, another method of physical control of microbes will be radiation. So there are two types of radiation that we need to be aware of. One of them, ionizing radiation. Ionizing radiation is generating ions, and in order of its penetrative ability, you can rank different types this way. Gamma rays, more energetic and more penetrating than X-rays, and X-rays is better than cathode rays. Okay. Now, what level of control you can achieve using ionizing radiation? Sterilization. It is a sterilizing method. Now, non-ionizing radiation involves UV light. And UV light is less penetrable than cathode rays. So this is, you have your line of ability to penetrate through the material. So UV light is disinfecting. Now let me talk about the very last physical method, and then I'm going to address what kind of questions you can be you can expect. So filtration. Filtration is ideal for fluids and gases. You can handle large volumes. It does not alter the chemical composition. Can be sterilizing. In case of filtration, it's basically the matter of money. How much money you are willing to spend on the filters and aero fluid handling systems. Does that make sense? Now, what you can expect, we're going to add some stuff here, what you can expect. First of all, I can ask you about what would you use for a particular thing. For instance, I ask you, what would you use to sterilize, that's important, sterilize medical instruments? You have very few options. Probably autoclaving. Can you use ionizing radiation? Sure. Do you want to throw them in the fire? Probably not. Just inconvenient. Does that make sense? What do you need to disinfect using physical methods? You need to disinfect something. You have options. You have UV light, you have boiling. I mean, if I ask you to disinfect water in the pool, you're not going to boil the water in the pool. You're probably going to run it through the UV light, for instance. Does that make sense? And I understand that there may be more than one option, and I try to formulate my questions and answers in a way that there is only one option in the answers. Does that make sense? Some of this stuff can be kind of questionable, like I ask you to sterilize food. You can argue that you can stick it into autoclave. You can. It will be autoclaved. It will be sterile. You can still eat it. It will completely lose its taste and appeal. Does that make sense? So this is why food is sterilized by gamma rays. It's a pretty common technique. Does that make sense? Now, broadly, what are the targets? Disinfect food. Okay, you say me, I'm going to pasteurize it. Fine, good for me. So, heat in general results in denaturation of proteins and DNA, okay? Extreme heat like autoclaving or, incineration just burned the thing to the ground. Autoclaving destroys all kinds of stuff. It destroys cell wall, it destroys cell membranes. Desiccation sucks the water out of the cells, they die. Okay? Radiation of all sorts, target DNA. Remember, we're talking about mutagenesis, double-strand DNA breaks. There you go. Any questions so far? Again, I know my questions, okay? And especially the application kind of stuff. If I ask you what would you do to, I don't know, disinfect a hospital room, you shouldn't look at physical methods at all. You're not going to boil the room. Well, technically you can throw in UV light and wait for it. Does that make sense? But like, is it reasonable even? Okay. Questions on this one?